is a general lecture on the clinical applications of molecular diagnostics. The objectives of this lecture are to identify the clinical uses and applications for molecular diagnostic testing. You should be able to list different types of tissues that can be used to isolate nucleic acids, to list advantages and limitations of molecular techniques, list the various types of molecular-based analyses that can be performed in a clinical setting, and list the technical considerations and prospects for using molecular-based tests in the clinical setting. So since we're going to be talking about molecular techniques, we have to mention DNA. And as you all know, DNA is the blueprint of living organisms. All nucleated cells contain DNA, which is the information that every cell needs to function. The problem arises when DNA becomes mutated or altered in some way, and these alterations lead to disease. And that's what we're going to focus on for many of the molecular-based techniques. Clinical uses of molecular diagnostics are varied, and you can use molecular diagnostics to diagnose a disease, to classify a disease, for example, staging a cancer, to predict prognosis of a disease, to monitor therapy, is that drug working effectively or not, and to assess drug sensitivity or resistance among pathogens. In the clinical lab setting, we can use molecular diagnostic techniques to identify genetic disorders, to find mutations in genes, to look at inheritance patterns of a disease, to look at neurological disorders, endocrine disorders, cardiovascular disease, to diagnose cancer, infectious diseases. So there are many ways we use molecular techniques or can use molecular techniques in a research and a clinical setting. So as we already said, applications in the clinical setting usually include cancer, inherited diseases, infectious diseases, as well as forensics. So for cancer, you can use molecular techniques to diagnose, classify, stage, and perform various types of tumor analyses. And in the clinical setting, in some settings, molecular techniques are being used to do all of these things with B and T cell lymphomas, chronic myelogenous leukemia, Ewing sarcoma, and rhabdomyosarcoma, among others. Inherited diseases, we can use molecular techniques, and molecular techniques are being used to diagnose, treat, and manage inherited diseases such as Fragile X syndrome, Leiden factor V, and hereditary hemochromatosis. Molecular techniques are used in infectious disease studies to monitor treatment, to determine viral loads, and to look at drug sensitivities as well as genotyping of a specific organism. So currently, molecular techniques are being used mainly for viruses and bacteria, but they can be used, especially in a research setting, for any infectious agent, including parasites, fungi, you name it. So for viruses, molecular techniques are being used for Epstein-Barr virus, cytomegalovirus, HIV, and human papillomavirus, among others. And for bacteria, Mycobacterium tuberculosis, Chlamydia trachomatis, and Neisseria gonorrhea. For infectious disease analyses, it's possible to use nucleic acids or molecular techniques due to several key principles. One being that all infectious agents contain nucleic acids. These nucleic acids include DNA and/or RNA. Also, different organisms have unique nucleic acid sequences, and you can use these unique sequences to identify what organism you may be looking at. You can also use these unique sequences to make probes, and we will talk about this technique later in the course. But you can really use these unique sequences to generate assays to diagnose infectious agents. You can use molecular techniques to perform various DNA identity procedures used in organ transplantation, bone marrow engraftment, paternity testing, various forensics analyses, and family analyses. You can analyze an entire family and try to identify biomarkers for a specific disease. The advantages of molecular diagnostic techniques are numerous and include that they have enhanced specificity and sensitivity compared to traditional methodologies. So, for example, older methods are more indirect methods where you're identifying an antibody or an antigen using various serologic assays such as ELISA techniques. The newer molecular-based methods are direct detection. Instead of waiting for an antibody to be developed at a high enough level to measure it, you can directly identify that nucleic acid, that DNA or RNA sequence. So it's very specific and very sensitive. Also, other advantages are that you can detect a single cell where you wouldn't be able to detect single cells using traditional techniques. You can monitor microorganisms, such as monitoring viral load for viral infections, and you can detect as few as 50 copies of DNA or RNA. Again, older techniques are looking at antibody production, doing CD4 cell counts to estimate viral load. So molecular techniques allow you to measure that organism directly. You can use genotyping, which is a great advantage because you can look at one single point mutation in a sequence, and then you can compare that mutation with the normal sequence. You can see what mutation works better with different drugs or chemotherapeutic agents. And when you know a mutation doesn't work with a certain drug, you can switch to a more beneficial medication. However, although there's advantages, there are limitations of molecular techniques. The first and foremost being that the cost is a bit high. It is coming down because molecular techniques are being developed more rapidly. There's kits available now, so the cost little by little has come down, but it is still high, especially for a lab to be using traditional techniques and have to convert to molecular techniques. It can be a bit expensive. The reimbursement may be low. You have to wait a long time before you actually get a return on your money when you completely switch to a new technique. Another major problem is false positives. When we're talking about amplification reactions later in the semester, if you contaminate that nucleic acid with an environmental source, you can then get a false positive reaction. So contamination can be a problem. Also, you might need additional lab space. You might need all new equipment. You have to really think about ways to eliminate or reduce contamination so that you don't get these false positives. And also, the testing is a little bit more complex. So you have to have appropriately trained personnel to perform the molecular-based tests. So our nucleic acid-based tests, any cell with a nucleus can be used to do molecular diagnostics on. So you can use peripheral blood with your white blood cells, you can use solid tumors. Any cell with a nucleus is going to allow you to obtain DNA and perform a molecular-based test. So blood. White blood cells have a nucleus, red blood cells do not. So blood is a great source for using molecular techniques, but it's the white blood cells in the blood that are going to allow for this. And all you need is a little drop of blood, about a dime-sized drop, in order to obtain enough nucleic acid so that you can do a molecular-based test. Sperm is a great source of nucleic acids for molecular-based testing. This is usually used for sexual assault or rape cases, and only 5 microliters of sperm gives you enough DNA. It's the equivalent amount of DNA to 50 microliters of blood. Now, the issue with a sperm analysis, especially in a rape case, is that you have to perform differential extraction. So usually you're going to have the vaginal epithelial cells of the victim as well as the semen from the perpetrator, and you really need to differentiate the two so that you know what is the DNA profile of the perpetrator and what's the DNA profile of the victim. Saliva, well, saliva itself doesn't really have DNA in it. However, saliva, being that it comes from the mouth, has oral epithelial cells, and that's where you get your nucleic acids. So saliva does contain cellular material, and you can use saliva from envelope flaps, postage stamps, cigarette butts in order to obtain enough DNA to perform molecular analyses. Hair. Now, hair is in itself not useful for molecular-based tests, but the hair follicle is. That follicle contains epithelial cells from the scalp, and those have a nucleus, so that's what you need. So if you find hair at a crime scene, it's not really useful for molecular testing unless it was pulled out from the root. Bone. Bone is useful for analyzing decomposed remains, and bone has been used to identify individuals from many, many, many years ago. So bone can be a really important way to use molecular techniques. Teeth, another excellent source of DNA, and again, like bone, remains there after the body is decayed and can be used, aside from doing dental examinations, it can be used for molecular-based analyses. Urine. Urine itself is sterile. It is not what you use for molecular-based tests, but urine usually contains your epithelial cells from the urethra, so you can obtain molecular material for analyses from urine. The various types of molecular analyses to look at various genetic disorders. You can do chromosomal analyses. You can look at monogenic or polygenic disorders. Your monogenic disorders are usually a disorder that involves only one gene, whereas your polygenic disorders are multifactorial. Many genes play a role in causing that disease. Also, you can look at mutations, and various mutations in genes will lead to disease. So looking at chromosomal disorders, you're really looking for any abnormality of the chromosome. So was there a loss, a gain? Is the chromosomal arranged abnormally? And when you have these abnormalities, it results in either too much or too little genetic material being made, and that can lead to various disorders. Monogenic disorders, your single gene diseases. These are diseases that display traditional Mendelian inheritance, and they're pretty frequent, 10 in 1,000 live births. Polygenic disorders, these are a little bit more difficult to analyze. Usually multiple genetic factors are involved, and they also don't commonly have your traditional Mendelian inheritance patterns. So some polygenic disorders, high blood pressure, heart disease, Alzheimer's disease, diabetes, and certain cancers. A lot of molecular-based tests are analyzing mutations or alterations in a genetic sequence. So mutation, when you refer to something as having a mutation, it means that that DNA sequence has been permanently altered. There's another type of alteration that we'll talk about later in the semester, and that's called polymorphisms. And they vary ever so slightly. So a mutation is a permanent alteration. You can look at different types of mutations, and usually when we're analyzing mutations to look at disease, we're looking for variations in the fragment length. So if you have a gene that's a certain length, you can use restriction enzymes or endonucleases that will cut that gene at specific sequences. Then you can run that on a gel and look for the varying lengths of where that enzyme cut the gene. And then you can get different patterns using that length variation. When you're looking at mutations, commonly you look at what's called hypervariable regions. And these hypervariable regions are areas of the DNA that tend to vary from person to person. Most individuals have identical DNA sequences among genes, but there are certain regions that are considered hypervariable. And what we look for are mini satellites and microsatellites. So mini satellites, or VNTRs, which stands for variable number tandem repeat, are repeat sequences that are usually between 10 to 100 base pairs in length that are going to repeat over and over in different numbers. Microsatellites, or STRs, which stands for short tandem repeats, these are usually di-2, tri-3, or tetra-4 nucleotide repetitive sequences. So these are little tiny nucleotides repeated over and over and over and over again. So mini satellites and microsatellites. There are several diseases that are due to trinucleotide or 3 nucleotide repeats, and these include Fragile X syndrome, myotonic dystrophy, spinal and bulbar muscular atrophy, Huntington's disease, and several others. So what you want to do when you're looking for these trinucleotide repeats, the number of repeats varies among the population. So the repeat is polymorphic. One individual may have a trinucleotide repeat that repeats 50 times. Another may have a repeat that it repeats 100 times. Usually these disorders are inherited from one generation to the next. So everyone has some of these nucleotide repeats, di, tri, tetranucleotide repeats. If you get a repeat number that's beyond what's considered normal, that is what results in disease. And we'll talk about this when we talk about inherited diseases. So mutations are highly clinically significant. You can use mutations as biomarkers. So you can use those mutations for diagnostic assays. Depending on the types of mutations that are there, the location of the mutation, which tissues are involved, but mutations are a great marker for disease. There's various types of mutations. We talked about structures of mutations, the types of mutations. There's germline mutations, and those are inherited from one generation to another. You can have acquired mutations. These usually happen, you have a normal sequence, and things such as UV light exposure can cause you to acquire a mutation. There are various types of mutations. Point mutations mean one single nucleotide has been mutated, and this could include a transition or a transversion. So a transition is when a purine converts to a purine, or a pyrimidine converts to a pyrimidine. A transversion is when a purine converts to a pyrimidine, or a pyrimidine converts to a purine. You can do direct molecular testing. Direct testing is where you're actually reading the DNA sequence and you're comparing that DNA sequence with the normal DNA sequence and looking for mutations or alteration in that sequence. The thing about direct molecular testing, you need to know what the sequence is in order to compare it. So that's one of the nice things about direct testing, but you really need to know that sequence before you can develop a direct test. Indirect testing is when you may not know the sequence, and what you need to do is then do linkage analyses. So this is where you're going to analyze generations of families that may have a disease and track specific genes and see if you can figure out what gene might be involved, and then you could do mapping on it. So the gene is mapped, but it's not sequenced. You're not comparing sequences, and we'll talk about all of these techniques throughout the semester. We can use molecular techniques to look at endocrine disorders, various endocrine disorders, cardiovascular disease. The problem with cardiovascular disease is that environmental factors can play a role in cardiovascular disease. So cardiovascular disease may be multifactorial. So there may be a genetic component, an environmental component, and all of these things may come together to cause cardiovascular disease, which makes it a little more difficult to study. There are monogenic cardiovascular diseases, such as familial hypercholesterolemia. And so that one is due to one gene, and 1 in 500 individuals has this disorder. Cancers, human neoplastic disease. Usually what happens with neoplastic diseases is you get accumulations of numerous somatic mutations. And the different types of alterations can be deletions, rearrangements, point mutations, and when you get an accumulation of these mutations, all of a sudden it generates cancer. Oncogenes are genes that are associated with cancer. You can have proto-oncogenes, which are activated forms of normal cellular genes, and that generates cancer. So a lot of times with cancer, what happens is there's a mutation that leads to enhanced gene expression, which is going to cause an overgrowth or an overproliferation of cells. There are also anti-oncogenes or tumor suppressor genes. So these result in a loss of function as opposed to a gain of function. Many times DNA is altered or mutated due to damage, and there's various agents that generate DNA damage, such as UV light. There are also individuals that are unable to repair that damage. So in our bodies, we have DNA repair mechanisms, numerous repair mechanisms. So right when our DNA is damaged by UV light, go out in the sun too long, we have mechanisms in our body that's repairing that damage. If your DNA repair mechanism is not working properly, that damage is not being repaired, and those mutations that are being generated can develop into a disease. Cancers that are familial or inherited include retinoblastoma, certain breast cancers, certain colon cancers. So there's many cancers that have a familial linkage. In order for a cancer to be considered familial, you need to see mutations of affected parents. So if an individual has a disease and you look at their DNA of the specific gene and you find a mutation, in order to say yes, that gene is the mutation, that's what's causing this cancer, the parents have to have that same mutation. You want to see germline transmission, a germline mutation that's passed down from generation to generation. You can identify the mutation in the gene and link that to being at higher risk for getting that cancer. You're going to see that same mutation in multiple family pedigrees, multiple generations, and you're not going to see that mutation in normal, healthy controls. Some of the technical considerations for molecular diagnostics. You need to have a target gene for a disorder. So in order to generate a molecular test, you need to know what gene or what piece of DNA is involved in that disease. You have to be able to obtain the appropriate source of DNA in order to perform the test. You have to understand the nature of the mutation that is causing the disease, and you want to make sure that the detection method you're using is sensitive enough to detect that mutation. So some of the prospects for a clinical lab to use molecular testing, you need personnel that's appropriately trained. You need to be certified. Both the laboratory needs to maintain certification as well as the personnel. You need to maintain quality control. There are ethical and social issues behind molecular-based testing. Some labs, high throughput labs, you may want to consider automated testing, and there's always advanced technologies being developed. So you want to make sure you identify the best technology for your laboratory. And as we already said, there are ethical, legal, and technical issues behind molecular-based testing. Cloning is one of the big ones. So whenever you're talking about molecular analyses, you're always opening up Pandora's box. So these are always things that you need to keep in the back of your mind.